Yeah, I think.
Yes, hello everybody. Yeah, hi everybody. I'm Shweta and welcome to the first RNA SpARC webinar. We will be conducting 2 more sessions like this in the coming weeks. So this webinar series is jointly organized by Ignite Life Science Foundation, IASC and NCPS. And for our first webinar talk, we have Srujan Merapally from CSCR-Vellore. His work is largely in the interface of chemistry and biology, with the major focus on lipid-mediated nucleic acid therapeutics and moving towards development of clinically viable applications for delivery of mRNA, siRNA, and plasmid DNA. And we will be having a Q&A session after this presentation. And please put in all of your questions for Srujan and for the panelists in the Q&A box. And we will be having Dr. Mrinmoy Dey from IASC to help us with the moderation or moderating the questions. And without further ado, let's welcome Dr. Srujan for this webinar on mRNA in lipid nanoparticles, Translational Science ne banaadi jodi. Go ahead, Dr. Srujan.
Thank you, Shweta. Good evening, everyone. I hope in this Saturday evening weekend, I try to entertain with some of, I prepared this webinar as a story kind of, so that you don't get bored. Yeah. So this outline of the talk is the first I'll explain about a shortened journey led to success of mRNA as a therapeutic molecule. And next, I'll introduce lipid-mediated nucleic acid delivery. And I discuss a few applications, mRNA vaccine development that everyone knows much. But there are many other applications with mRNA that, like as a protein replacement therapy or other editing applications that I'll briefly explain in upcoming slides. And finally, I'll conclude my presentation with the current challenges and future perspectives. So a shortened journey led to success of mRNA as a therapeutic molecule. So when we are talking about mRNA, if we don't talk about its predecessor, pDNA, it would be really an injustice to the field. So the first molecule as a therapeutic molecule, a nucleic acid is considered as pDNA. That started with gene therapy. The definition of gene therapy is very simple. Using genes as medicines to treat diseases. So pretty straightforward, like when there is a gene is mutated, then you just supplement the gene and that's, the problem is solved. So, but that's not that simple as one thought. There was multiple challenges and still the world is, struggling to have an ideal vector and an ideal therapy for these disorders. So the aim of gene therapy is to eradicate the symptoms rather than, eradicate the cause rather than the symptoms. And the first choice was the viral vectors, as we all get infected with virus frequently. So that's what they, initial efforts were made, and then the viral vectors are made retrovirus, then lenti, and then AAV. But these viruses have immunogenicity and hepatotoxicity. And there is a lot of challenges in the vector development of these viral vectors. So among these viral vectors, adeno-associated virus is one of the most successful vector in pDNA, plasmid DNA-associated gene therapy in vivo. Among these vectors, actually, the critical challenges are that, AAV, when it is like any vaccine kind of, once the virus is injected, the antibodies are developed. So vector re-administration is not possible. Hence, the early stage intervention, if we do, then once the liver is developed, then again vector dilution happens and we need to inject again. So early stage intervention is not possible. That too particularly for those who are with genetic disorders. And the vector re-administration is not possible because once the antibodies are developed, we cannot re-administer it. And there in our CMC research, we have serosurveillance, then we found that over 80 to 90% of the patients have multiple seropositive to these antibodies positive to multiple serotypes. So that's one of the major challenges also we have in this field. So that's the reason why these gene therapy, like, I just to compare because it grabs attention immediately. So that the space tourism, if you take, this is what the data from, I think the SpaceX, this $0.2 to $0.3 million they are charging, and then the gene therapy products. If you say, Hemgenix is the recent launched gene therapy product for hemophilia B, which charges $3.5 million, and Onpattro, Zolgensma is $2.1 million, and Glybera is 1.6 million euro. So these are pretty expensive, like almost 2 to 5-fold expensive therapeutics, and some 10-fold expensive therapeutics. So that's what these gene therapy is a luxury affair. So that's because of the challenges with the vector development in particular. So obvious choice goes to the non-viral vectors, and these non-viral vectors are the lipids, polymers, dendrimers. They are less efficient in pDNA, but efficient in delivering RNA, and they have broader applications, easy to scale up. So hence, among all these lipid nanoparticles emerges as a potential alternative. And this is the mechanism how the lipid nanoparticles enters the cell and delivers the cargo. So through endocytosis, it enters, and endosomal escape that happens there, the release of cargo into the cytoplasm. If it is a pDNA that has to be entered into the nucleus, and there it has to go back to the cytoplasm. Again, the mRNA transcribed, and that has to be into the cytoplasm. So the critical challenge is in the delivery part is the nuclear membrane. Probably we all might be using the transfection reagents. These transfection reagents can efficiently deliver pDNA. You hit 293, you may get 80-90%, but why you are not getting in the same kind of effectiveness in the primary cells is because in cultured cells, the nuclear membrane is compromised. When there is a cell division is happening, then when you treat the lipid nanoparticles and there is a cell division happening, the nuclear membrane is also compromised, so that there is a possibility of entering possibility of entering these lipid nanoparticles into the from cytoplasm to nucleoplasm. Whereas when the nuclear membrane integrity is not compromised, there the chance of getting into the nucleus is a challenge. So that's the reason where this was a bottleneck in the field for several decades. However, this lipid nanoparticle based delivery started more than 4 decades back, but the efficiency was very less, and it was not relevant to the therapeutic fidelity. There, the story was stuck there. But when it comes to the mRNA development, there you don't need to deliver into the nucleus. So it is just into the cytoplasm. So the route was shortened for mRNA. That made this entire field not a clinically viable field. So that clinical needs also, it's not just confined to the hereditary disorders, but broaden the scope to genetic disorders, acquired and infectious diseases. So these mRNA the discovery from 1961 to the advanced therapeutics in 2021, recent approval for COVID-19. The chronology is that in 2005, both Kathleen Carrico and Drew Weissman identified that these base modifications are critical for the higher translation and making it a therapeutically viable product. And in 2015, almost a decade after, there is a significant development in the mRNA technology, where trialing came with CleanCap technology, and N1-methyl pseudouridine was further more effective. So all these contributed to expedite the development of mRNA-based products. So this is one of the first evidences that how mRNA was directly injected through hydrodynamic injections into a mouse model. So these hydrodynamic injections, little force we need to inject. We can do it in animals, but not in the humans. But this is just a proof of concept way back in 1990. This is the first attempt to make use of mRNA as a therapeutic molecule. But without any delivery vector, it is not possible. So the delivery vector, lipid nanoparticles, contributed to the success of mRNA. So these lipid nanoparticles also, in the chronology time, if you take 4 years later, like 1965, these are started as liposomes. And the first product was, preclinical study was done in 1995 on the influenza virus. And in 2000, the lipid nanoparticles the liposomes are approved as a drug product. And the first clinical trial with mRNA encapsulated, that's with BioNTech's cancer immunotherapy. So these are where there were different paths of development. Actually, lipid nanoparticles were designed to develop, they were ionized. That time it were called cationic lipids or ionized lipid, I can say that, because positive charge of amine is constant there. It's quaternized amines. Whereas in ionizable lipids, there is a tertiary or secondary, so that it can be ionized to positive charge. So that those are the primary differences, but the principles remain the same. So these lipid nanoparticles were developed to deliver pDNA. However, the efficiency was less, so the field was struggling. So then the mRNA contributed, like in 2015, it's made for each other, that the pair is made. There it helped both emergence of lipid field and also emergence of mRNA as a therapeutic molecule. So from then there are a lot of, this field expanded to protein replacement therapy, vaccine development, gene editing, CAR T cell therapy, antibodies, and iPSC generation. And next, I'll briefly talk about the lipid nanopart-mediated nucleic acid delivery and how that is important. Now, most of us, we don't know that these lipid nanoparticles, those particularly in the field, they think that it just started when it is, mRNA technology is developed. But it is not true. There were multiple attempts. And when C. Mello, Andrew Zephyr got the Nobel Prize in siRNA, then Philip Sharp and colleagues started Alnylam Therapeutics exploring siRNA as a molecule. Because siRNA is a small duplex DNA, and it's just 21 mer RNA. Sorry, it's just 21 mer, so that it cleaves mRNA. So this one, this is the first Onpattro. The Onpattro is for H880R amyloidosis, and this is a drug was approved. The lipid nanopart, the siRNA against H880R, the gene is encapsulated. The mRNA is encapsulated in the lipid nanoparticle. And Givlari for acute hepatic porphyrias also been approved. These two are Alnylam products. And when the COVID came, the emphasis moves more on the mRNA technology, and the Pfizer and BioNTech got the first approvals, which the mRNA of spike protein of SARS-CoV-2 is packed into the lipid nanoparticle system. Typically, these lipid nanoparticles have a charged lipid. It's a positively charged lipid, whether you call it as a cationic lipid or ionizable lipid. And then it has two colipids, and that cholesterol or any steroidal that imparts some rigidity to the lipid nanoparticles, steroidal structures, and other colipids such as DOPE or DOPC that impart some fluidity to the membrane of the lipid nanoparticles. So this is a few examples like ionized and ionizable lipids and helper lipids are being used in clinics. So typically, these lipids have a structure of hydrophilic head group, hydrophobic tail, and connected with a linker functionality. So there are multiple attempts on the hydrophilic head groups, like how they function, like tethering. If you like tether a ligand for targeting, then that becomes a targeted lipids, like galactose. When you tether it, that goes to liver through a shallow glycoprotein receptors. It's a ligand for the SGPRs. And when it is tethered, mannose or quinic or shikimoil ligands, that goes to the dendritic cells are macrophages, because these receptors are overexpressed. So the linker is the functionality, like it is a breakable functionality. One can say like either ester, amide, or multiple other functionalities are there, but these are most prominent, esters and amides. So we, from 2005, my lab has been I've been working on this field, and I've been trying to understand how best these structures influence the transfection efficiencies of the nucleic acids. And we found that even you change a subtle variation, there is a subtle variation, you flip the bond, there is a significant effect on the transfection properties, because of the exposure of positive charge is compromised, and that led to the change in the arrangement of the liposomes, and that eventually affected the transfection properties. And also the hydrophobic chain, which is already people just thought that it is just embedded in the lipid core, so that it may not have a significance, but that's not true, because these fusogenicity is imparted by the asymmetry, and the asymmetry plays a very critical role that we, in multiple attempts, we demonstrated that it's, the imparting hydrophobic asymmetry improves the transfection properties. So I'll just quickly explain one simple story, like all we bath with soap. Now, what is soap? It's just a saponificated vegetable oil. Like you take out the glycerol from it and make the fatty acyl things, that becomes a soap. So we what we thought, like it has a lot of asymmetry. If you look at the if you just characterize the soap, you have multiple fatty acyl chains from myristoyl to linoleic acid. So we started exploring that asymmetry. That's why we call it a green transfection. We just made a couple of lipids. With one stretch, one reaction, you get 10 to 12 lipids that we characterized, and we evaluated the transfections, and we found that it is showing better than the any individual. Like here we used palmityl as a control, because palmitic percentage is higher. And then also the steroidal molecule. Always definitely cationic lipids play a role, or the ionized lipids play a role in LNP transfection, imparting transfections to the nucleic acids. But we thought like it's not just alone the cationic lipid, but the overall the membrane structure, since the asymmetry is imparted in it. So that's the reason we thought like if we just take another steroidal moiety which has naturally close to the structurally close to the cholesterol. That's what we chose long back in 2017. We did this work, and we chose spirozolines, a tomatidine, and that's actually it has a slight membrane perturbing properties. This is plant defense mechanism against the host, like the pathogens. So it permeates the membrane of the eggs of the parasites. So that's the thing that we adopted, and we chose it. And we found that when we doped the tomatidine into lipid nanoparticles, the transfection efficiencies went up. And the same thing we also did with the steroidal sapogenin, the diagenin. There it also modulates the endocytic pathways, like the entry route. And interestingly, when we the reagents are like for a specific purposes, like either for plasmid DNA or for siRNA or for mRNA or for protein. But this is one such because this has a endocytic modulation properties. Now it could deliver all three components like plasmid DNA, RNP, and mRNA. The one single transfection reagent. We could do that. So the design principles, what we get out of it is that understanding the molecular architecture of the lipid is very, very critical. And naturally closely mimicking ionizable or ionized lipids are critical for the nucleic acid deliveries, because when that ultimately gets broken down, it should be broken down like what we have a natural phenomenon, so that when we need to re-administer the lipids, this is not the safety profiles are not compromised. So most of the designs are just confined to the random design, like putting too many amines or too many hydrophobic chains, split it into multiple things. But when it comes to the safety part, so far nobody shown a proper safety profile of a lipid nanoparticles. Lipid nanoparticle is not an inert particle that we are thinking, it's not just a mere carrier. It has a lot of influence on the not only just the delivery, but also after the safety profiles. So that's why it is another drug. In my opinion, if we are going for a clinical evaluations, the lipid of the novel lipid also should be evaluated as stringent as a novel drug. Otherwise, we don't know the real safety profiles. And the exploration of co-lipids for enhanced transfections, like how best we can pack that lipid nanoparticle system, depends on the components that we are using. So there are multiple other novel components. We can closely, like how we did for the tomatidine or the spirozolane, we can also look into those like nature, and for each specific application we can design specific excipients of the lipid nanoparticles, so that it with a minimal cationic lipid, it can pack it and efficiently deliver it. The composition of lipid nanoparticles in lipoplexes should be optimized for cell-specific applications, because we cannot generalize. Each cell has a different component, or tissue-specific. I can say that I'm going to talk a little more in detail in my upcoming slides. So now I'll briefly explain about mRNA applications. Like everybody knows vaccine development. Like even we were doing for actually protein replacement therapies way back in 2017. But when COVID came, we also had to start our things for mRNA vaccine development. And before that also we were working on DNA vaccination, where we were developing shikimoid mannose mimicking ligand lipids, because it is more stable in the endosome. So that's we call mannose or the shikimoid or quinoid ligands. We tether it, and then we do, mostly these are ex vivo, and also we have done in vivo. Ex means we transfect dendritic cells, and then we infuse those, and or you directly inject with MART1 antigen that we used to work on. And when COVID came, we started this novel design of the lipids, and similarly these lipids, because it's a shikimoylated, this targets the dendritic cells through mannose receptors, which we evaluated that these shikimoylated lipids enhance it targeting of the lipid nanoparticle, delivery of lipid nanoparticles into APCs and DCs more than 2 to 3 folds than the normal LNPs. And we also have our own design of mRNA, which has our own UTR design, and also the spike protein is also designed differently. In our case, I'm not going to explain in detail, but this is why I'm explaining and just showing this, because the design of mRNA is also very critical. So here, the reason we chose a different, because we try to shorten the length, that helps in packing more molecules in one lipid nanoparticle, and also mRNA synthesis. The shorter mRNA has a lesser double-stranded RNA production in IVT. So it's easy to purify. So that's where we chose without compromising the activity, and we demonstrated in a preclinical mouse model that this has a better immune responses. And we cut story short that these lipid nanoparticles showed enhanced antigenic response against multiple COVID SARS-CoV-2 variants, because when the more spike is delivered, then the amount of the cross reactivity increases. So that's what with serological data we published another article on that. And also now the world is just not looking for one vaccine for all. We have these RSV is one such. Recently, couple of days back, there is in the news that Moderna got a vaccine FDA approval for RSV, but there is there is co-infections like RSV and COVID. This is very common. It's happening. This is now highly prevalent. So there is an attempt is being made from a group, Wu and et al. They demonstrated that having there's a fusion antigen, fusion mRNA from RSV and spike mRNA from SARS-CoV-2, that they made mRNAs and packed into the lipid nanoparticles, and they optimized the immunization regimen, the first dose with spike LNPs and the next doses with SFLNPs, means spike and fusion LNPs. That showed a good, because this is a very critical to overcome antigenic competition. Their strategy they helped in overcoming the antigenic competition. So this is because the success with mRNA for vaccine development is immediate, because you don't need multiple administrations. Hardly you may need 1 or 2 administrations. One booster dose and other booster dose, or even after that, intervals are pretty large. So, but when it comes to the protein replacement therapies, there the challenges lie, because the protein replacement therapies you have to repeatedly inject. That's where the critical challenge lies, and these and the safety also is very, very critical when you want to repeatedly infuse some exogenous material. So we chose the protein replacement therapy, the hemophilia, because it's liver is easy organ to target, number one, and number two is that it is a rare disease, so that the category of getting into the approvals will be easier. And the second is that the success also, you don't need to 100% replace factor 9 here, even factor 8 or factor 9. You just need to replace even 10% is good enough, 5% is good enough, to stop bleeding. And if it is 10%, the patients are normal. So that's one fantastic thing with this thing. So that's the reason we chose hemophilia. And there are multiple short-term protein replacement therapies are there, but they are very expensive in the clinic, and that's not available in the market. And AAV gene therapy, as I mentioned earlier, is that it's pretty expensive, like $3.5 million. Nobody affords in India. So overall, more than 80% of the population is without any therapeutic, like just either clotting factor concentrates or the recombinant proteins. So there, the challenge is there, like AAV's gene therapies once for a lifetime, but still that's a star quoted, because there are certain clinical trials which is showing reduced expression of the proteins, therapeutic proteins, and the 2 shorter half-life. Weekly twice you have to infuse the recombinant proteins or clotting factor concentrates. So either you have a long term or you 2 short term. So the always middle path is the best path. But Indian philosophy is that's what we chose. Nucleic acid, based therapeutics are the best choice for this thing. And already there are like Anpatro with siRNA, but the challenge with siRNA is that siRNA itself induce some toxicity, hepatotoxicity. These are there. So the long-term use of these kind of drugs are also still debatable. No, however, futiziran is a fantastic drug for hemophilia in our phase 3 clinical trials, conducted in CMC, and we, our clinicians found that just one injection is good enough up to 3 months, 2 to 3 months. So we, inspired with that one, we thought of developing an mRNA-based therapeutic, and this, like what we found is that, previous literature, this is what Inder Verma's group with Arcturus Therapeutics, but the expression, this is the first protein replacement therapy published for, but the expression time is only 7 days, and also same thing happened with Moderna. So there's factor 8, non-factor 9, both, they were showing up to 7 days. So mere increase of, twofold, and thinking that infusing, a lot of exogenous material with, lipid nanoparticles may be a real challenge, to translate this technology into the clinic. So the effective strategy must have a long-term expression of the functional protein. So how do we do that? How do we extend the half-life? So we thought like, developing a liver-specific system and make use liver as a depot to that, a sustained release. So instead of just one burst release, we thought of a sustained release. And second is optimizing mRNA structure for higher stability. So we worked on these two aspects, and we found that I explained you like SGPR receptors, which, and the liver is the major organ for many biochemical functions. So there are umpteen genetic disorders originated from the liver. So that's what, once we develop and deliver this technology, that can be a platform technology for many liver disorders. So this is what the futiziran is, like the GalNAc they used, and here we used a modified beta-D galactose with a different triazole linker attached to it. So this is what Jennifer Doudna's and Intellia Therapeutics, they started a clinical trial. They packed mRNA, Cas9 mRNA into the lipid nanoparticle system, and they injected into the patients with HATTR. They edited HATTR gene, that helped in reducing the burden of the mutant protein, so that their amyloidosis is ameliorated, and there is a significant reduction in the total the protein levels of HATTR. So this there they used endogenous targeting that I'm going to explain to in the later slides. And there is another, I forgot the name of the company, but this is another mRNA technology-based company. They started a hybrid technology called Hybrid mRNA Technology Delivery System, HMT, where the polymeric micelles and lipid nanoparticles are combined. And that helped for the sustained release of mRNA, and that shown this ornithine transcarbamylase. They showed up to multiple, the efficacy is there up to multiple weeks. But the challenge was that, like, these, because the GalNAc, galactose also induce a toxicity. So that if it is a beyond certain level, and when there is a repeated administrations, that becomes even more critical. And then they used GalNAc polymer. Probably that could be the reason they got high inflammatory responses in this. So that's thinking these things in the mind. We thought like, we will develop the same galactosylated lipid, but here we are using a combinatorial delivery, like the ionized lipid and ionizable lipid. The ionizable lipid is the targeted lipid, that gal triazole that we name it as, and the cationic lipid or ionized lipid is amide 16, MEOH, means methyl and ethyl hydroxy. So we made multiple formulations with that, and then we evaluated physicochemical characterizations, and then we looked at the mRNA transfections, and we found that this Galena 5 is showing a better transfection, and because the MFI is high, when 1 to 3 is almost the same, but MFI is, for 5 MFI is more higher, mean fluorescent intensity. So we chose Gal 5 and Galena 5, and we evaluated their stability and the physicochemical characterizations further, and they showed higher stability up to 30 days, and they are stable even in serum presence. And to know the sustained release, the long-term expression, we just mimicked the liver, liver extracellular matrix has collagen. So we did a collagen hydrogel embedded HEPG2 with the lipid nanoparticles encapsulated with EGFP mRNA. And we could see up to 14 days the expression. However, that was less in the 14 days, but there was expression. Then we injected into the animals with a label dye, and then we could see, you could appreciate the liver specificity of the galactosylated lipid nanoparticle system. So this is where we could see the galactosylated lipid nanoparticle. But specifically, sorry, this is where it is into the galactose, the liver, and this is into the liver, lung, and spleen. So the specificity is imparted with the galactose ligand. And then we moved on to the screening of the base modifications. We looked at multiple base modifications and evaluated both adenine modifieds and guanine, cytosine, and uracil modifications, all the available modifications. We systematically screen because there is no available literature in this thing. So interestingly, we found that when we also looked at the yields, like how much is per reaction it's happening. So N1-methyl adenosine and 2-methoxyguanosine we couldn't get any good yield with that one. So that's one we found from these things, like some modifications may not, but 5-methyl cytidine showed a better yield compared to the other base modifications. So overall, the yield is not significantly affected with the base modifications. And then we looked at the translational kinetics, like how these base modifications really fare in the in cellular conditions. Like when we delivered it, the GFP positive cells, we didn't have a big difference, particularly with pseudouridine versus N1-methyl pseudouridine and 5-methoxyuridine. But we could see an appreciable difference after 48 hours, and the mean fluorescent intensities are higher for N1-methyl pseudouridine. And very interestingly, this the fold change and the looking at the mean fluorescent intensities, the mean fluorescent intensities are pretty higher, like 2.5-fold higher compared to the unmodified, or compared to the unmodified. So this is for N1-methyl pseudouridine, and the similar trend, this is in endothelial cell and this is in hepatic cell lines, and the similar trend was observed. However, this is even more evident with N1-methyl pseudouridine. So that what we overall conclude from these experiments, that the base modification also play a critical role in the mRNA stability within the cell. So if you see these base modifications, because of lack of time, I'm not showing all the data. In HET293 cells, you may not appreciate the difference, but you could appreciate the difference in hepatic cell lines. So these base modification of this factor 9, that what we all did, like we engineered with albumin UTR, because albumin is the largest protein produced in the liver. So the UTRs are robust. So we thought and then we engineered factor 9 with albumin UTRs and N1-methyl pseudouridine modification. So we found that these modification and this albumin UTR showed a higher translation and stability in the factor 9 expression. Similarly, in concurrence with our studies recently, few weeks back, BioNTech also demonstrated that albumin UTR, cloning with albumin UTRs in mRNA, showed a higher translation and longer expression. So then we did animal studies, and then we injected into the animals, and we looked at different time points of the factor 9 expression, and we found that this is almost up to more than a month, slowly started expression, and it lasted up to 36 days. Then we evaluated the safety profiles and all the safety, multiple injections also we evaluated I'm not showing all the data, but they found that this is pretty safe. So overall, we developed a liver-specific nanoparticle system, and we engineered, mRNA with the liver-specific design, and the Galinas showed the functional protein expression up to 30 days. So this serves as a platform technology for long-term protein replacement therapy for liver disorders. So the world is looking beyond the liver, like not just confined to the liver, but also to the multiple other organs, lungs, spleen, kidney, heart, and brain, so on. So these things actually, multiple attempts have been made, but so far, even with the viral delivery, if you take for the Zolgensma, which aims to target the muscle cells, with an AAV targets muscle cells, but the major depositions also there in the liver. So that's why their hepatotoxicity, and probably that's the only drug available on the earth with more mortality, but still it is considered because more than around 10% mortality, I can surely say. But since it is used because there is no option, the patient had to die without any therapeutic or other option available. So, but if you consider the other things, like these are really challenging things. Crossing the liver is one of the humongous tasks that still we need to look into, like what are the best strategies and how we can reduce. We may not eliminate 100%, but if we reduce the burden, then that becomes a success story. So the targeting depends on the passive targeting, where you look at the physical properties of the nanoparticle, size, shape, and stiffness and surface charge. Like those are the one. It is called passive targeting. The active targeting is like you tether a ligand, like how we did for galactose or the mannose. You tether, and that goes into and delivers the molecules, the receptor-mediated endocytosis. So that's one. Called as active targeting, and endogenous targeting, where the lipid, when it is designed, it doesn't have any such targeting ligands, but when it is injected into the body, that has a interaction with the serum proteins, and that serum proteins direct the cargo to a specific organ. So that's called endogenous targeting. So I'll just go, in the interest of time, I'll just briefly go through the previous examples, like how people have been working on to develop these strategies for muscle. Like there is a group from Akitsu. They used a TCL053 lipid nanoparticles, and these could maximize the delivery of both LNP, the CRISPR, and the ASO2 MDX mice, muscle of MDX mice, and rest root. There is also one interest in the CAR therapies being booming these days with mRNA Fab CAR in vivo, like one injection that will give fibroblast associated protein. This is for the CD5 targeted lipid nanoparticles they used to restore the function of the heart. This reduces cardiac functions. So then another story on the brain, like how tryptamine conjugated lipid nanoparticles could cross the blood-brain barrier and delivers the cargo into the brain. And also, interestingly, not only just that, even to the placenta, these lipids are found to be effective in delivering into the placenta. There are another passive targeting that we are talking about, that's called selective organ targeting, SpARC lipids. This is from Seegert group. They developed, they demonstrated how positively charged, negatively charged, and neutrally charged lipids have a different biodistribution profiles. And how they demonstrated that positively charged goes to lung and liver, whereas the neutral goes to the spleen and heart kind of these demonstrations that they show me. And overall, these examples, we could design the lipid nanoparticles in such a way to fit the need of the therapeutic application. These are few examples from our lab and from the other labs. And this is a summary like how the passive targeting helps, and there is a comprehensive strategy like what we anticipate that the things which involves in the passive targeting plus active targeting and the help of endogenous would make a better targetability overall and reduce the other off-target depositions. So there is one from our collaborators group where they demonstrated that directly compared with the lipid nanoparticles with the lipoplexes, because many confusions or many people think like these lipid nanoparticles are the only things, but before that it used to called as lipoplex when lipoplexes are complexed with the mRNA or the nucleic acids. So they how this microfluidic mixing helps and the demonstration. They looked into, they have a direct comparison neck and neck between lipoplexes with LNPs, and they interestingly there is no big difference they found, and the lipoplex showed some better delivery efficiency in NK cells. Again, these are cell specific, they were engineering NK cells. And more interesting thing is that unmodified mRNA showed higher translation than modified RNA. So far, we were thinking like, if you don't modify mRNA, it won't work. But even the unmodified mRNAs in NK cell engineering, it worked. It worked better than modified ones. So that's an interesting concept they are developing. Like how an intrinsically active lipid nanoparticle design will overcome the modification. So that's also another strategy is being employed in this field. So the conclusion is that there is still we achieved success with mRNA for vaccine development, but still there are multiple things need to be optimized for multiple other applications, such as if it is for an editing applications, you don't need for a long-term expression where you shorten the expression is the better result that you may get. So you need a very short expression, so that you tuning the mRNA is very critical, like how best you can tune your mRNA. And second thing is that the specific delivery of the lipid nanoparticles. As I discussed earlier, that with these principles we can have a better design in future, and I hope that there will be multiple applications in next decade. There will be several products, mRNA-based products for protein replacement therapy and antibody therapeutics. With this, I conclude my presentation, and I'm open to I thank my lab members and funding bodies, collaborators, and core facility of CSCR, and I'm open to the questions.
Thank you, Srujan. It was a great presentation, and I think we will open the questions. But before we start, Dr. Mrinmoy will be taking the questions. So he is from ISE and he's a chemist, and he largely focuses on synthesis of various kinds of macro and nanomaterials for various applications, including delivery and antimicrobial research. So without further ado, let's go to Mrinmoy Day. And I have one question. So right now, all the mRNA that you were looking at is linear, right? What about circular mRNA?
Yeah, actually, the circular mRNA, actually there is an interesting article being published by very long back when I was doing PhD, Dr. Shantanu Bhattacharya from IISc. There, the group evaluated the packing of circular nucleic acids versus linear nucleic, means pDNA versus linearized pDNA that time. So they found that when the molecule is circular, you need a lesser amount of cationic lipid because the packaging is even. But when it comes to a linear molecule, that becomes a little challenging and you need more lipid. So considering that point, what I think if it is a circular RNA, the packaging would be good and the expression time would be longer. But the challenge is how best we can scale it up to a circular RNA is a thing to consider.
Thank you, Srujan. Please, Mrinmoy, go ahead.
Yeah, Srujan, nice talk. So already a few questions are here.
Yeah,
and, first we will go with those and then we will go for more general questions for discussion.
Sure.
Thanks. The first question I can see from Vishwesh. So Vishwesh's question is that how long is poly A tail that you could incorporate in your optimized mRNA?
Yeah, it is like same 120, 60 plus 60 with a linker function.
So it's the same basically.
Same 60 plus 60 plus linker. No, 60 and then linker and then 60.
So is the optimized length, or there
Actually,
are some possibility of optimizations?
See, actually in mRNA, there is a lot more to be optimized. In fact, even there is a very recent paper is being published with a modified adenosine, that has a better stability. And we cannot just say that that's what even when we looked at base modification also, these are not just a generic. I know 120 length cannot be a generic to all cells. Say suppose if you are aiming for hepatic, that would be 120 or maybe lesser. We have to have a screen, like a thorough screen of it. Then only we get to know which is the best length.
Okay. Yeah, so Vishwesh's question is very similar in that direction, that is that the optimized length, and what is the kind of that can be addressed for that intercellular stability of mRNA? Is that length also helping that intercellular stability?
Definitely.
Yeah. Right. I think Vishwesh, you got the answer. The second question is about that delivery possibility. So the question is that did you ever explore the possibility of delivery of LNPs in intracellular parasites? And if it is so, then what are the challenges related to that? So,
intracellular parasites we haven't explored so far. So
okay,
the thing is there are some attempts have been going on for multiple anti parasitic effects with these mRNA based therapies, but I really have no first-hand experience on that. The critical challenge would be after delivering into the cell, how we can target the replication of the parasite. That makes, I think in that case, it doesn't too much a different story, like whether it is a vaccine or that. The challenge is to deliver into that particular cell type is the challenge. Once it is there, whatever, if we design targeting that, say suppose, there may be you take hepatitis virus. So that hepatitis, you target, if you target that sequences, so then that, with Cas9 mRNA, that can be editing also, you can do parasite editing. So mRNA technology for this thing, mostly it is being used with the gene editing approaches now. So this is another interesting field evolving with gene editing plus mRNA plus antibacterial or antiviral therapies. So in that case, how we design the targets and how best we can deliver into the targets, that depends on, in my opinion.
Yeah, so I mean, this is kind of a question that, I mean, in one slide you have shown that mechanism, right? So how the LNPs is kind of the internalizing and then delivery of the mRNA in the system. So before basically cell targeting, you can modify the LNP for specific target, right? That target could be like whatever the possible targets you have shown, like it could be a cell target like dendritic cell or particular organ or something like that. But after the delivery, after the reaching there, then you do not have that any targeting control. That's the point you want to mention.
So after that one, so there are two things. In case the scenario one, if the parasite is not replicating, it's just been stagnant. If I understand the question correctly and I'm answering, or you please get back to me if I'm wrong. What I understood from the question is that if there is a parasite inside a cell, how we target that parasite? Is that one? Then the thing is like the parasite, if that remains as an inert, like it is not replicating further. There is no growth of the parasite. Then we need to target that parasite. But usually these are the things like, say, suppose even if you take a plasmodium, they replicate in the cell, use cell machinery to replicate. So if you their things are exposed, their parasite may not be in the intact. Their things are exposed at one point of time. So at that point of time we can catch and target that particular sequence with Cas9 mRNA or base editing mRNA. You mutate those sequences and make that parasite non-replicable, then that serves. Actually, the world is going towards that direction now.
Okay, yeah, nice. So I will move to the next question. This question is from Durga Rao. Question is that what is the frequency and position of pseudoridine in the mRNA and the effect of pseudoridine in codon specificity? Yeah.
No, it is completely replaced. There is no other uracil. This thing completely pseudo
replaced. Yeah,
100% replacement.
And okay, yeah, fine then. Yeah. Another question, I don't know from whom. It's a general about how can mRNA in lipid nanoparticle be used in plant genetic engineering, which I'm not sure of though.
I really don't have any answer for this because I'm not into the plant domain. But whether the overall mRNA technology, certainly we can use it because we can use through gene gun or electroporation. But usually plant membrane is pretty different from the animal plasma membrane. Right. So it is really not designed for these things, but I'm not sure whether we can use or not. But yeah, definitely mRNA technology can be used, that I can say.
Yeah, so point is that, I mean, whatever my thought process is that I actually don't know what are the challenges for the plant genetic engineering. I mean, do you need this LNP platform or probably that thing? Because LNP platform is a different reason, which is basically in that initial introduction you mentioned that why you need LNP. That requirement is there for the plant system that I don't know. If it is, then maybe it will be helpful.
Yeah, we have to even look into that, whether we can explore this in plant genetics also.
Sure. This is the question so far I can see. I think more questions are here. Again, it is from unknown. So question is that, is it possible to add tissue-specific receptor with LNP to increase tissue-specific delivery? Like, for example, isialoglycoprotein, I don't know about that. Receptor is a liver-specific membrane, whatever you use.
Yeah.
Yeah, but you have mentioned some of those things like a tissue targeting LNPs, right?
Yeah,
Maybe
yeah.
you can
That's what
explain
even in my
further.
latest slides I mentioned, I explained about the targeting, like how best we can target either through passive targeting or active targeting or endogenous targeting. So one example I demonstrated that one with the galactose ligand either to the lipid, and that delivers cargo through asialoglycoprotein receptors into the hepatocytes of the liver. So this is one, like, one strategy, and the next strategy is like endogenous targeting, like what Jennifer Doudna group Intellia Therapeutics used. So they are designed in lipid nanoparticle system in such a way that it interacts with the serum apolipoproteins, and that apolipoproteins direct to the receptors, and that also, because there is multiple receptors are expressed on in a particular cell type, and particularly hepatocytes is a large is a big cell, and also have a larger component of the liver. So that's another approach people used to explore the targeting. That is called endogenous targeting. Either active targeting or endogenous targeting, we can deliver the cargo tissue specifically. But the challenge is like liver is whether you have a ligand or not. The more thing goes to liver, even in my the experiments that we found, whether it's a targeted and non-targeted, it goes to liver anyhow. But the thing is how well you can reduce the distribution into the other organs. So the galactose modification only help to restrict that into the liver, not into spleen or heart, the lung. But whereas non-modified ones went to the lung and spleen. So we can stop the liver beyond the liver, the molecule, the lipid nanoparticles delivery. The liver, imparting liver specificity is well understood. It is very, this much before that there were, like Mitsuru Hashida pioneered this one. He had at least galactose-related liver targeting more than 30-40 publications, even 2010 back on these things. But the major challenge is how we overcome the liver is the challenge. So that's where the world is stuck.
Yeah. So the point is that, I mean, most of these things will go to liver anyway, but it is like a restricted to the liver, or it is after liver, it can be spilled other places or not. That is the main target here, right? Whenever you are thinking about the liver targeting.
Yeah. But if you are planning for other extra hepatic delivery, means, say suppose for spleen, there we need to have a different strategy where the liver deposition should be minimized or very, very minimal, whatever. Because the therapeutic cargo, you can maximize it in the target arm. If it is deposition, there may not be a major complication, but say suppose the efficiency, if there is a liver half in the liver deposition, the efficiency is only half. So for that, we need to increase the dose. Increase the dose, then again the toxicity will increase. So it is actually a good balance, like how best we can overcome this.
Okay, great. Thank you, Srujan. We'll go to the next question. Yeah, that previous question is from the Krishna. I just read that message through. So next question is from Dr. Rush from IIHVB Bangalore. He has a couple of questions. So the first question is that how can the stability of the mRNA LNP in vaccine formulation be overcome? So it's a big challenge, but you can give some light.
That's what the world is working on. Like, it depends. The stability depends on the lipids that we are using, and the lyophilization conditions that we think of. Like, we can the cryoprotectants definitely help in playing a major role in it. And it is important, like that during the lyophilization, it's like you are removing the water from the sample. When that is happening, there will be a residual, and that if that is crystalline, that becomes like it's like a small knife kind of to the mRNA, so that when you are thawing it, that things that will rupture or that will affect the mRNA stability. So for that, how best the lyophilization conditions need to be optimized with the lipid composition optimization is critical.
In this regard, I have one question. So, whatever the literature supports that this kind of a stability, again, always it is like case by case, like one mRNA versus another mRNA.
Definitely.
And then different composition change, different structure of the lipids generally change. Is there any structure activity relations anyone work or any is there?
So far, to the best of my knowledge, there is no such.
Yeah.
Because each mRNA has its own stability. And second thing is that when you are that depends on the length of mRNA, and there you are packing how you are packing the system. So the multiple things play a role in it. So I don't see any generic answer for this, one fits for all. And there is no certain structure activity relationship studies on this thing so far, to the best of my knowledge.
Right. Okay. Second question is that is there any difference in the action of MRP LNP across that BBB compared to other organs? So,
pardon, I didn't get MRP.
Yeah. So, is there any difference in action of those LNP, mRNA LNP in BBB, blood-brain barrier, compared to other organs? So, how that thing can be differentiated?
No, here how the targeting ligands, how virus infecting in multiple areas. Like if you see, the virus, it has a simple genetic material, either with a protein coat or like a lipid coat, like that they have. Same thing. Similarly, the targetability depends on how your ligand, which ligand it has. So again, that also depends on the serum protein interactions. These two contribute to that, to overcome these LNPs to the blood-brain.
Yeah. Okay.
That's what the attempt with the tryptamine and from Arvinda Chaudhry's group also, they shown with other ligands, tethering other ligands. So those direct the cargo.
Yeah, most likely the question is towards this in direction is that whenever we are using LNPs, mRNA LNPs, compared to other organ, is there any effect, or better to say side effect to brain, like what it will reside on brain or the decomposition around brain? That is probably the question.
No, usually these LNPs won't cross blood-brain barrier that easily.
That's the main point.
That's the reason we have been getting injected with means this Western world now we haven't. But even in fact, even we take the anti-cancer drugs, Doxil, other anti microbial liposomal formulations are also there. They are if that effects are there, then it's not being used in the clinics, right? So there, it's not that easy to cross blood-brain barrier until and unless if you have a defined targeting ligand towards that.
Yeah, I think crossing the blood-brain barrier is not so easy, particularly using the size of the mRNA, some size of the LNP is generally used. So, yeah. Next question is from Dr. Ramdas, but it is very generic questions. How are you purifying mRNA?
No, there are two things. In what we do is a cellulose column. We just use cellulose filter columns to do that. We eliminate double-strand RNA with that.
Yeah, there are those things are there. Okay. The next question again from Vishwesh. This is about the specific patterning and multivalency of ligands on the surface of the nanoparticle has been shown to be important when it is come to binding and cellular uptake. As for example, how a viral particles uses multiple ligands to target a cells, okay, natural system. When lipid has a phase segregation tendencies, then have you observed the surface of liposome under such conditions and whether it affects transfection?
Yeah, that's a fantastic question. Yes, no, the multi-targeting, there are people are started exploring it, like how this thing happened. But no, so far we haven't done multi-targeting, to be honest. We only chose one. But what we chose in that single targeting, the critical thing is to getting internalization. So escaping from the endosome, how best that escapes. So that's why we use a triazole moiety there, because that has through proton sponge effect, that drags water molecules in the endosome. There the endosomal disruption happens and that releases the carbon. Yeah, but we haven't explored multi-targeting, like how mimicking a virus is. But that's an interesting concept. We also would like to explore in future.
Yeah, this is always like this uncertainty in this direction. So a lot of structure and all those things people worked on to find it out. That's where the different models, design, structures of all lipids are coming in this publications and
Yes,
market.
yes,
Yeah.
absolutely.
The next question is from Dipesh. Is it possible to use mRNA to other purpose to repairing damaged organ?
So the mRNA, now that's what even the liver regeneration, if you take our antibody therapy, there are multiple other it's not just one, the vaccine or protein replacement therapy, but there are multiple other things are also being explored in the current. I gave some examples like how we can treat, means there are attempts to, including actually because of the interest of time, I couldn't show much of my data. Like even we have the muscles, like how we can regenerate the muscle, because the muscle degeneration happen in older people. So we are working on that project. So we now have some interesting data on that, like now lipid nanoparticles with mRNA. So delivering into the myotubes is a challenge. So how we overcome it. So there are multiple stories, but I just stick to the two complete stories. But yes, we can do it. Now that they're not only just confined to this simple vaccine or protein replacement therapy, it is to multiple other, to treat. It can be an antiviral, it can be an even anti-aging, you can say, or liver regeneration, multiple effects. Like we can do that.
Okay, I think that's the all questions so far I got. I have one thing, whichever you mentioned, which I have not thought earlier. This is you mentioned about that drug effect of the lipids itself, right? Drug effect of the lipid.
Drug effect, yes, yes. No, what I mean to say is that the problem, if you take the lipid design, so even we really don't know. ALC is one which is in the, just for one injection, ALC was there. But you look at the structure, the biodegradability of that structure, if you consider, it is so complex. I don't know how, if we really use for that effect, that for a protein replacement therapy or other applications where we need a multiple administrations. No, it is really not possible. No, we need to look at how that may not have. I'm not saying that it may have that complication. Yeah. My opinion is that when people are using any chemical component has some effect on the system. It is for sure, 100%. Even sodium chloride has that weight. No? So we cannot think that something which is an ionized or ionizable thing, which doesn't have any effect on the body, number one. And when we are using in larger quantities for longer time, then definitely it is as a drug. So this is not just a mere vector. What I mean to say is that we should think the lipid nanoparticle design itself, because even there are a lot of crazy things are being happening in this field. Like people started designing lipid nanoparticles. Even in some high-profile journals, you can see like because there is a struggle, fractured tail, like they have a small dual tail with a small hydrocarbon chains. So that they've been doing multiple chains and making it into a lipid and showing efficacies. But ultimately how they degrade is also important. And when we are looking for the other applications, hence that I brought it like drug.
Now, this is a very important direction, because always we are thinking about that whenever our main target is that, or whenever we are studying also, that main target is that what is the level of expression, what is the extent of delivery and all those things.
Exactly.
That time we always use control as just a lipid as a control, and we just go ahead with that. Now we never generally look back what happened with the lipid, or is there any effect. Now, my point is that this is a very interesting point in the sense that if there is any effect, so how you are going to screen that? Like, I mean,
no, but like any that's what I brought up in the issue. Like if a new drug is there, like how we do its PK studies. Probably the pharmacologists can do these things, and with HPLC after injecting into the blood, like multiple intervals, they can take that and look at the profile. Like it's not that easy. Like even simple metformin, we can say simple metformin mechanism was unknown. Like how the metabolism happens after. So it's not that easy. But what I mean to say is that the stringent toxicological and safety profiles at least need to be first documented.
Okay, great. So there are no more questions I can see. So in that case, I will ask Shweta to take over from here.
Sure. Thanks, Mrinmoy. I think this was a great discussion session, and I think we will thank Srujan for this great presentation and all the participants who came up with some amazing questions, as well as the moderators and the organizers, the team from NCBS for enabling this webinar. And everybody, please look out for another notice for the next webinar, which will come out soon. And we will see you again for a next exciting session. Instrumentation. Thank you.
Thank you.
See you, everybody.
Thank you, Srujan.